In the germination process, the role of the seed is that of a reproductive unit. Seed as reproductive units will have different requirements for germination depending on the species. In this lesson, we will look at important endogenous and environmental requirements of seeds for germination. Seed maturity is a universal requirement for germination. In seeds, there are two types of maturity. One is physiological maturity, and the other is harvest maturity. Recall that following pollination and double fertilization, the zygote and the primary endosperm undergo controlled cell division and differentiation towards developing into a mature embryo and endosperm. Now, we may not see the actual division and differentiation of the cells, but we can see the development of seeds based on changes in physical size. Take, for example, the grains of sorghum, barley, or wheat. After double fertilization, you can see how the grains feel every day, and this grain feeling coincides with cell division and differentiation happening within the grains. When the grains are filled and the funiculus detaches from the ovary wall, a seed has achieved physiological maturity. During this stage, the seed reached the end of dry weight accumulation but has not yet undergone the controlled process of drying that is supposed to partially arrest metabolic processes within the seed. During this stage, seeds can still have 20 to 50% moisture content per dry weight, depending on the species. Despite the relatively higher moisture content, physiologically mature seeds are able to germinate under favorable conditions. In rice, for example, physiological maturity is reached 8 to 14 days after pollination and subsequent fertilization. Physically, the grains will still be green, but they are already capable of germinating because the embryo is already fully formed and the endosperm fully developed. Harvest maturity is similar to physiological maturity in that the seeds have already reached the end of dry weight accumulation. That means the glucose have been polymerized into starch, the proteins have been packaged in protein storage vacuoles, the lipids have been partitioned into oleosomes, and the minerals have been bound as crystalline phytates. The major difference between the two stages of maturity is that harvest mature seeds have already undergone a controlled process of desiccation. That brings the moisture content of seeds to 10 to 15% dry weight of the seed. The low water content partially suspends the metabolic activities within the seed, allowing it to remain viable during extended periods of dormancy or quiescence. At harvest maturity, you can imagine seeds being in a state of sleep. So how does seed maturity affect germination? Seeds of most species are capable of germinating at physiological maturity. Some very good examples of these species include rice, wheat, and tomato. However, there are some species like soybeans, for example, that requires harvest maturity. The ability of soybean seeds to synthesize enzymes necessary for germination is developed during the slow process of seed maturation and desiccation. Without undergoing the drying process and achieving harvest maturity in that process, soybean seeds will not be able to germinate. These panels show the physical difference between a physiologically mature and a harvest mature rice grain. In the left, you can see the grains and seed coat of a physiologically mature rice. You can see that they are still green. In contrast, the harvest mature seeds, pictured in the right, have a beautiful golden seed coat. The green color of the grain and seed coat in this physiologically mature panicle is an indication that they are not yet fully mature and that they have not undergone the process of drying, which is a key step in seed development. Despite the green color, the seeds are fully filled and will be able to germinate under favorable conditions. This table shows the effects of seed maturity on the germination of sow thistle and Canada thistle. These plants are largely considered weeds, but they have also been used as native ornamentals. Now, the first column on the table shows the number of days after pollination, which represents the different stages of maturation of the developing seed. If we look at the sow thistle column, we can see that few seeds were able to germinate only after 4 days after pollination. Maximum germination of 83% was recorded for sow thistle after 9 days of pollination. In contrast, Canada thistle requires at least 6 days, right here, after pollination for seeds to germinate, and a maximum germination of 90% was observed after 10 days of pollination. Overall, this data tells us that plant species will have different requirements for seed maturation level for optimum or maximum germination. Aside from maturity, which is an endogenous property of the seed, the environment also plays a critical role in the ability of seeds to germinate. From this slide onwards, we will look at various environmental stimuli that are necessary for seed germination. Let's start with water. Water is a basic requirement for seed germination. When seeds mature, they undergo a controlled process of desiccation or drying that brings the moisture levels in the seeds down to 10 to 15%. The low moisture level partially suspends the metabolic processes in the seeds so they don't germinate unnecessarily. To reactivate these metabolic processes, seed moisture content needs to be raised up to at least 50% of the original dry weight of seeds. An example of this metabolic process is enzyme activation that is necessary to break down, transport, and use stored reserves in the seeds. The water requirement for seed germination is not the same across plant species. Under low moisture content, for example, germination in most plants will be impaired because of the insufficient amount of water available. Remember that water is necessary to reactivate the metabolic processes in the seeds, and this include enzyme activation that is important in the breakdown of storage reserves. Without sufficient water, enzyme activation might be impaired. For other plant species like forages, germination is still possible even with low moisture. Some examples of species that require minimum water for germination are wild rye and tall wheatgrass. With high moisture content, seed germination might also be inhibited, just like in the case of sugar beet, corn, and dwarf bean. In extreme cases, high moisture content can induce seeds to germinate while they are still attached to the mother plant. This condition is known as vivipary. These are pictures depicting vivipary in rice, corn, tomato, and strawberry. Again, vivipary is a condition where seeds germinate while they are still attached to the mother plant. In this picture of rice panicles, you can clearly see the radicle protruding from some of the grains that are still attached to the panicle, and these are indicated by the red circles. Here, and you can also see it here. In this panel, not only can you see the roots, you can also see the shoots coming out from the grains that are still attached to the main panicle. In the case of corn, tomato, and strawberry, you can see the green shoots of the new emerging plant. This condition is most commonly induced by combination of warm air and high humidity or moisture content in the air. This is something that you don't want in your crops. So imagine harvesting corn with shoots coming out of the kernels. You will not be able to sell your produce, and this means losses. Seeds of some species, especially those of non-domesticated plants, can be extreme in their water requirement. Some rice relatives, for example, especially the land races and the wild species, can germinate underwater. The rice plant depicted in this panel here, and also here, are actually land races. I think it's very obvious from these pictures that this land race or land races are able to survive flooded conditions, including during germination. The low oxygen levels imposed by submergence in water of seeds have been shown to stimulate coleoptile growth. Now, modern rice cultivars, just like the rice that's depicted in this panel, can also germinate underwater, but they are usually weak. Aside from water, air is also a key environmental determinant of seed germination. Air is composed of approximately 20% oxygen, 79% nitrogen, and 0.03% carbon dioxide. During germination, the seed undergoes respiration in support of cellular activities that will lead to the development of initial shoot and root structures. Now, respiration is an oxidative process that requires adequate supply of oxygen. That means, for a seed to germinate and sustain respiration, it will require oxygen. Most seeds require oxygen concentration that is close to the oxygen concentration in ambient air to germinate, and that oxygen concentration is 20%. In some species like wheat, sorghum, corn, sunflower, and oats, lower concentration of oxygen reduces germination rates. Seeds have various chemical components that serve different functions. The stored reserves in seeds, for example, are primarily for the nutrition of the growing embryo. Alongside this, There are chemical components, especially in the seed coat, that inhibit the germination of seeds until conditions are favorable. These chemical inhibitors are leached out mostly when the seeds start imbibing water. Oxygen has been reported to reduce the endogenous levels of these chemical inhibitors. With reduced levels of oxygen, higher levels of germination inhibitors are maintained in the seeds. The non-leaching of these inhibitors may prevent seeds from germinating. This table illustrates how air in the form of carbon dioxide and oxygen affects the germination of oats. In the first column, we have carbon dioxide concentration. In the second column, we have oxygen concentration. The third column lists the germination percentage of oats in response to variable carbon dioxide and oxygen concentration. Now you can see that when the oxygen concentration is equivalent to the oxygen concentration in ambient air, which is 20%, oat germination is 100%. As the oxygen decreases, as you can see in this table, and the carbon dioxide concentration increases, the germination of oats decreased. The observed decrease in germination is proposed to be due to the anoxic conditions created by low levels of oxygen. With low oxygen concentration, ethanol is generated, and ethanol is toxic to the seeds. Some plant species actually germinate better when oxygen concentration is below 20%. Examples of these species include lettuce, onion, we have the Bermuda grass, and also the cattail.